Hi, Han, can you hear me?
Hi, I'm great.
Hi.
We're a little bit early, so we're just gonna wait for
Yeah, 2 minutes earlier.
Are
So
you
how are you?
in the office?
Yeah, I'm in office today.
Hello, everybody. Welcome for joining. There was somebody in the Teams link, so I'm a little bit late, but I see he's now also here. So nice to meet you, Rowan. Just checking if everyone is here. I see many new names. Is someone from the team of MycetOS already here? Yeah, I see that MycetOS is joining now.
Hi, everybody.
Hello, Matt.
Hey, Matt.
I think we're all there. I'm just opening MycetOS to get the agenda. Because I noted that we were convening, so
I'll be taking notes as we go.
So first of all, welcome. Maybe because there are some new faces, it's good to make a round of introductions. So I would like to start a meeting with that. My name is Wendy van der Sande. I'm an associate professor at the Erasmus MC. and I'm in charge of the biological screenings of the compounds selected by the MycetOS community. Next to my screen, I see Ma. Ma, could you introduce yourself?
Hello, everyone. My name is Yingyi Ma. I'm a PhD candidate from Wendy's Group. I work for drug screening in vitro and in vivo. Thank you.
And I see the next one next to me is Hang. Hang, could you introduce yourself?
Yep. Hi everyone, my name is Hung. I am from the University of Sydney. So I'm actually a PhD student, and I am doing synthesis for the Mycetoma project. But I'm also, on the other hand, I'm also doing some chemistry educational research as well as part of the other part of my PhD. But yeah, that's just a quick intro for me. So right now, I think it's just me who's will be sort of like corresponding and working on the project at UCIT, but hopefully we get some more people to join us from UCIT in the next year or so.
Okay, thank you, Hang. Next to Hang on my screen is Louisa. Louisa, would you like to introduce yourself?
Hello, hi everyone. I'm Louisa. I'm here to kind of replace Ben, although I don't think I can do that very well right now. But Ben from the NCEs. So my name is Louisa. I have organic synthesis and medicinal chemistry background. I've been working with medicinal chemistry for neglected diseases for the last 4 years in the project with the NCEs here in Brazil. And for the last 5 months, sorry, I've been with the team from the NCEs. I'm based in Campinas, but I work for the Latin American office in Rio. And yeah, I'm excited to be part. So now I'm going to be the point of contact for at the NCEs for the Open Synthesis Network and all that open source work that we have at the NCEs. So yeah, I'm excited to be here, and yeah, let's talk.
Okay. In my screen after Louisa, there's Anthony.
Hi, I'm Anthony. I'm someone who's basically self-taught. I do some of the modeling and some of the like GitHub maintenance and stuff like that. I'm not affiliated with any university or anything, but I'm part of the group anyway and taught myself. So it's been a pretty interesting ride.
Okay, thank you. And then for me, I hope I pronounced this correctly. The next name in the screen is Saina.
Yeah, it's Saina. Hi, I'm Saina. I'm a second-year student, pharmacy student at UCL. And I was just really interested in the project, and I got invited by Professor Todd to join this meeting today.
Okay, welcome. We always like it
Thank
when
you.
young students join.
Thank you.
And then in my screen, the next one is Bernard.
Yes, hello everybody. I'm Bernard Piersack from the University of Bayreuth in Germany, and I'm also contributing some test compounds to the project.
Thank you. And then after Bernard, in my screen, there's Rowan.
Yeah, hi everyone. Great to finally meeting you all, and I'm here to learn a lot more about the Mycetoma project. I'm a natural products chemist, and I've been at Griffith Uni in Brisbane, Australia for almost 20 years now. I'm the manager of Nature Bank, which has large extract and fraction libraries that are screened for various human diseases. And I've got pure compound libraries as well. And hopefully some of those compounds will head their way to Netherlands in the next little while to be screened in your project. So very much a newbie to this project, but willing to learn and hoping to contribute. Thank you.
Thank you, and nice meeting you. And then next in line is Matt.
Hello there, everybody. I'm Matt. I'm a professor of drug discovery at University College London. And with Wendy and Ben and a few others, we started MycetOS a few years ago now. Forget how many years ago it was, but a few years ago now we started MycetOS. Welcome, everybody.
And then the last one in my row is Dimitri. Dimitri, go ahead.
Hi, I'm Dimitri. I'm a PhD student at UCL, and that's doing chemistry in mycetoma.
Okay, thank you. In my list, I got everyone. If I forgot someone, please call out your name, then you can introduce yourself. Then if not, then I would like to go to the next part of the agenda, and that's the outstanding items from previous meeting. From the outstanding items, the first item was that Dimitri was to summarize in a new post the latest in vitro and in vivo data from August and September in advance to the next meeting. But I assume you needed my help on that. Were you able to do that? I did look in the manuscript and I made some corrections there.
Yeah, so my reference file was the presentation from September by Mimi. So the blue and the orange box. So this kind of covers 71 compounds from biological assay from July till September that was done. So I can share probably the screen and show you the potency quickly, just the main points, I guess.
Yeah, I know that we had some issues with the potency because the volatile oils generated, or the volatile compounds, so we had to dilute before we got a clear hit. But yeah.
So just a quick reminder from the presentation that was given in September from our collaborators from the Netherlands. There were like two sets of compounds, not sets of compounds, but two sets of structures that were tested, and they were divided into the blue box, which mainly consists of synthetic compounds, and the orange box, which consists of natural compounds plus 4 drugs. Yeah, 3 antifungals and 1 zoledronic acid. I'll post this on GitHub so that everyone have the scheme available to them. But just the main points are there were compounds from University of Sydney, from UCL, from Germany, from Bernard, and from University of Otago. If we're talking specifically mainly about the potent ones, I have highlighted them in the green, the ones that gave IC50, and those are mainly from synthetic compounds, 1,4-nattoquinone and 1,2 plumagin. They gave like very good results, and they proceeded to the IC50. As far as I know, and from what I can see from the mycetoma spreadsheet, none of them have been able to prolong viral survival. Wendy, correct me if I'm wrong, but that's or the data is still missing there, or is it final?
Well, the IC50 is only done if they are inhibiting. So when compounds are not inhibiting in the start, then we don't determine the IC50.
What about the larval survival? Because I couldn't see like any data on that. Like, were they proceeding to the next?
Larval survival data that takes a lot of work. So we only did larval survival for the Fennery moths to finalize that part. These IC50s are also relatively high. So that's a concentration we would not be able to reach in the larvae anyway. So those would not have been selected to go further, simply because we cannot reach that high.
Yeah, but they were like the most potent, like out of the entire like synthetic library. And thiazoles, 2 that stand out is like MyOS 3.15 and 3.17. also had like a very good inhibition both at 125 micromolar, and IC50 was slightly less better than the quinones. With regards to the rest, what I can say, the blue box, synthetic box, the Ortega compounds mainly had like moderate potency, but like none of them like proceeded to the next stage. But it's a bit strange because, for example, 396 has a very high good potency results. It's 4 and 3 micromolar at 3%. Survival growth rate at 100 micromolar, but I can see like any IC50 data, so I wasn't sure. But I checked like the whole list, so this is pretty much up to date.
Yeah, we didn't finalize all the data yet because Mimi only had 2 months, and first she needed to learn the assay, of course, and then screening. And Ma was busy with the larvae experiments, which are season dependent. So I really need to go through the list, which ones are still needed to finalize for IC50 and which ones not, because
so hopefully this will be helpful, just to go through the list as quickly as possible, because it's now like all organized in a coherent way. And we can always update this further. University of Sydney compounds in that batch from July to September, there were only 3 compounds. They were not potent, inactive, unfortunately. The ones that came from UCL were mainly negative control compounds. We predicted that they would not be active. For various reasons, absence of the pyridine ring, which coordinates the heme iron, or absence of the halogens on the phenyl ring. But they were inactive as expected. There was only one compound that gave inactive results, was the pyrrolidine analog. And the last one was for the phenarmols. The last compound for series 1 phenarmols within the range was 61/1, which was also highly potent. Those were the 5 compounds from the batch. Natural compounds, none. Well, probably I guess it's a bit too early to say like final outcomes, because maybe we need to go through this again. But apparently none of them will proceed, will not get IC50 values as well. Same for the 3 marketed antifungals. They gave like moderate potency, but did not reach the required threshold as well.
Yeah, I will have to go through those compounds one more time just to see what the final outcome was. But yeah, I simply didn't have the time for it yet.
I have a question for these compounds here, because I think Mimi has presented them in one of our last meetings, and you told us, or told me, that there was some damage to the microplates because the compounds were tested in such a high concentration that they attacked the microplate plastics. Is this true, or are these new data? Is this new data from
we retested several of the data, and only the data which were okay are uploaded. We had some issues with indeed some compounds which were turbid upon storage. So those ones, I think it was a problem with freezing. So we did them at took the original ones, diluted again, and retested.
Okay, thanks.
Just quick questions. So this sheet, and is this all of the recent compounds that have been evaluated?
71 compounds, July to September.
Okay, so this is everything.
Yeah, 71 compounds.
And because they're all on MycetOS code, I'm assuming that they're all in the master list.
Yes, it's that file that I used as a reference. Wendy knows what I'm talking about.
All right, so they're all in the master list. Is great. So in the master list or elsewhere, how are we capturing compound provenance? So are we capturing somewhere simply where the compounds have come from?
Yes, there is a special line which usually says
in the master list.
It has the name of the person who sends in the master list.
Okay, fine. Because that's important. It's crucial that is always filled in when something is added to the master list, some idea where the compound comes from, because this is an open source project, so it's very important that if people contribute things, that those contributions are recognized and written down. Obviously, all these compounds here are, the compounds on this sheet are from different series. So ultimately, we need to group the compounds together by series type, and post the picture of those in the relevant repository, right? Because obviously only a few of these are actually phenoromols. There's a bunch of other compounds and other series there. So the maximum usefulness will come from making sure that we're comparing like with like, so that we can learn from which compounds are which. So fine to dump everything in one diagram in the issue that we're currently using, but ultimately the pictures for the different series need to go in the different repositories, so that we keep track of what's new.
Yeah, so I'll break this large file into subgroups based on their series.
Yeah, and then just post them to different repositories.
Yeah, also answering on the question, as you can see, like on the blue box, so for example, the first row, so it has the MycetoS code, but then I also for the readers, for them to be easier to understand, I've put the second code. So for example, you can straight away tell that it's like U015 is from Andreas compounds or Tago. And that's how they you can track them like in the Mycetoma spreadsheet as well. The only problem is
what we're going to do here is to look at the actives and inactives and then try and figure out what's good, right? So some of the series 5 compounds that are shown there are structurally very similar, but there are quite significant differences in potency. Do we have a sort of agreed color coding for things like this? So green is for proceed to IC50, but do we have a kind of agreed set of oranges and reds that we can use to further triage things a little bit? If everything's black, it can be quite difficult to spot patterns.
So for example, for Keroxime, I used that color system, but that's something that we use for the paper usually. So green is potent, or the one that gave like good results at 25-100 micromolar and good IC50 results. Inactive, or even those with moderate activity, are still the ones that do not go throughout the threshold barrier. Personally, I usually put them as red. We used to have orange as a medium potency, but it's not very helpful. Maybe it's helpful for the SIR analysis in the paper, but still the end point of this is that they don't proceed to the next step. They're kind of like still regarded as inactive compounds. Green,
because I guess the numbers are the main. Yeah. The numbers for in each case they go between 100 and 0, right? The numbers underneath the log D.
Yes, correct.
So I mean, if there's a way of having a sort of red, orange, and green thing to give us a bit more granularity on what's looking promising versus what's really great versus what is completely useless, it would be kind of helpful.
Yeah, unfortunately with our assay, it's not always clear, like 50% inhibition, it's not always clear. So it's best to just say growth, no growth. So beneath 20% or above. What we could do with the yellow or orange is in vivo and in vitro potency green, in vitro only yellow, something like that. But with respect to the vitro activity, yeah, I wouldn't make it a gradual scale, because especially when you're inhibiting it, the 50% threshold is not very consistent. So it could be 50% in one assay, and then your replicate, it could be 70%, and the other replicate, it could be 30%.
That's what we're usually seeing in the mycetoma spreadsheet, like before you calculate the mean average, the numbers differ. That's exactly how it is. The thing that you just mentioned, Wendy.
Yeah.
Would it be useful to have a heat map of the compounds, something? Because if we're talking about at least like 3 color coding system, then it's like a heat map, something like that.
Yeah, but based on what Wendy just said, I'm not sure it's justified. Because it seems like it's, you know, promising and not promising. Are the two, for in vitro anyway, are the two classes. So not sure.
I would
Just
personally
trying to
do
visualize
red
it for
and green and red, to be honest, based on what just Wendy said. But we can adjust it.
Yeah, for now I would leave it green and red, and we could always look later on if there's more nuance. Because if we are going to look at different species as well, then you might also say, okay, if it's inhibiting more than one causative agent, then it's green, something like that.
Sure. Am I good to go with rearranging the screen based on the subgroups, or are we still missing some essential data? Because we need to make sure that they're like up to date. How are we gonna resolve this?
Well, I think according to the current data sets, because Ma, I'm looking at you, we didn't finalize the IC50s for those compounds yet with the blue box, did we? We still need to calculate the data.
With
From
the mimes, I don't remember it. I think.
Okay, we will discuss later, but as far as I knew, she did do one run, which still needs to be repeated, and we need to calculate the overall data because we usually don't rely on only a single run.
Sure. So my suggestion would be I will regroup the scheme now, and then we will update if we have new data. Just somehow we need to make sure that we communicate this and don't forget about that in the future. So we'll do it like that for now. Yes. Yeah, so this part is probably of the report is finished, so the potency results. Can I quickly give the UCL update just to finish up on my part?
Yeah, that's okay. For the outstanding action points for your side, it was also the EpiChem library update for closing the SAR gap. So I guess that's also part of your update.
Yes, it was posted on GitHub before the meeting some time ago.
Okay, yeah.
Sorry, I just didn't open it. So it looks like so the scheme. So we discussed this with now before the meeting. So what still needs to be done? So I did the SR gap analysis and I found additional compounds to what Frank has suggested before.
Dimitri, you're breaking I can't hear you very well. So could you speak a little bit closer to the microphone?
Yeah. Is it better now? Are you hearing me better now?
It's still a little bit difficult to hear from my side, but I'm not sure if others experience the same problem.
I think it just sounds a little bit, you sound a little bit softer than before.
It's a bit strange because it didn't change anything right now. Just put the computer closer. Yes, so the EpiChem analysis from my side is finished. So I found like additional compounds. I tried to group them based on their relevance in terms of what we're looking for. But the next step which needs to be done, so this has been posted like before the meeting, like more than a week ago. So the next step like in this analysis would be match pair analysis because we need a bit more clearance. So the compounds that, so what I mean by that is the compounds that we see in the, on this list, on the scheme, tested against the tested mycetoma compounds to see which match pairs do we have. So this can be like addressed like more in depth, in a more in-depth way. Apart from that, the list is complete. It's on GitHub now. So maybe Hank, you can, if you have time, you can have a look, and then we can combine your list with mine.
So the thing we need here, we need a subset of these compounds, which we select for testing, so that we can answer specific questions. So in each case, the gap that we have were some questions that were posted previously about whether or not certain parts of the molecule were important, or whether variation would be tolerated. So in terms of the first box, for example, I think the idea there was that we were asking whether or not substitution on the pyridine was tolerated. And so we need to pick a molecule in that top left box, for example, which is structurally very similar or identical to an existing molecule, where the only difference is some substitution in the pyridine ring. And then if we can identify that second molecule, which is almost identical apart from that change, if we can identify that pair, then we can elect to test one molecule which addresses one or two molecules or something which addresses that question, so that we can answer that in the paper. And similarly, top right, we need a molecule that is identical to something we already know about, apart from some change in the core group, the central group. Or if we don't have one, that we pick two molecules in that box that are identical apart from that change. So in each case, we need this distilled down to a smaller set of compounds that will help us answer those questions for the paper. We don't need to test all of these.
Yeah, I think I can help with that as well, because I've done some of the stuff that we found in terms of like the SAR gaps. I should be able to perhaps just have see if I can have like a smaller subset of this, because I can say some interesting compounds from what I see here as well. So
yeah, so it's a list of sort of 4 or 5 questions or whatever it is that say, is this region of the molecule, can we vary this? What happens if we do this? What happens if we do this? In each case, those need to be gaps in the SAR, and we nominate 2 or 4 compounds, something of that kind, which if we can test them, we can answer those questions. That's the point. But if we want, we need this for the paper, right? So if you want to include this in the paper, we have to get on with this, because it's going to take a while to get the compounds from Epichem to Wendy and to test them.
Yeah, so testing means testing in vitro first, or testing also in the larvae, because the larvae will have to wait till next season.
No, it's vitro. It's fine in vitro. It's just for the SEO.
Because in vitro, if we do the simple screening, the high-low, because the number of compounds, I only see the top. But if it fits in a 96 well plate or half a 96 well plate, then this amount doesn't seem that it's not doable.
It's going to be reduced for sure. We will pick the best ones. It's just I went through the entire like Epichem library, picked like my selection, but of course.
No, what I meant is that the amount seems doable for me. So if you need to restrict because you think you need, due to the number of things we need to screen, because when we screen this in high-low format first, that means that we screen 1 or 2 times 96 well plates. So that's not the issue. It's more the issue if we need to screen it in the larvae, then the more compounds, the more work. But in vitro, 1 or 10 compounds or 100 compounds is usually not that much more work.
Yeah, the reason really is that we're requesting something here from Epichem based on good faith, right? So if we request a compound, someone's got to actually go in and get the compound in Epichem, and this is not their core business at all, right? So if we're able to reduce things down for their benefit, I think that would be sensible. And we only really need to test a subset of compounds in order to answer the questions for the paper. So at this stage, I don't think we need to test them all for the purposes of the paper, because, you know, I mean, a lot of these compounds are remarkably similar to each other.
Okay, that's fine.
Yeah, let me have a look at this first, and I'll probably just get back to I'll have a look, and then I'll report back.
The original ChemDraw file is available, was posted as well, so you can open it as it is now.
Yeah, so just put this one on me for now to have a look, and I'll go back once I have some time.
It's on GitHub. Sure. Yep. Thanks. Yes, so probably that's it, I guess.
Okay. So those were all your updates, weren't they, Dimitri?
Sorry, Wendy. One more thing, based on a quick update, UCL update based on the library. So we have another batch of compounds ready to go for analysis. So the question is whether we should expand more on this and send it later, or are we more or less ready to go quite soon? Personally, I would do more compounds because there are a number of interesting entries that I would still like to try. So, for example, during research, I came across one compound which is of high interest, I think, to the project as well. The marketed drug oxyconazole. As far as I know, it's the only catoxime that has been marketed like as an antifungal. So I'm not sure. I need to look through the mycetoma spreadsheet. I'm not sure whether it has been tested against mycetoma before. Maybe it has. But the proposal here would be to make something similar to the oxyconazole, but with our pyridine scaffolds, with our classical scaffold for Series 6. But this is something that we'll decide at UCL when we have a look at the mycetoma spreadsheet as well, and maybe a bit of SAR in literature. That would be, yeah.
I don't know by head if this oxyconazole was tested, but I know there are plenty of azoles in the library, the MMV library, because also the recently FDA approved azoles are there. So yeah, you could take a look in the database if it's there.
I would personally do it because it's quite interesting. But so the idea here would be, if it has been tested, then we compare the biological assay of this UCL analog and then compare. If not, then maybe we would we'll be able to get oxyconazole. It's not expensive. It's only around like $8, if I'm not mistaken. So it should not be expensive, and we can test this as well. Okay. So that would be the suggestion. It's of particular interest because, especially for Series 1, because it's the only catoxime. And we, what I also found, the E isomer of this is being used as a marketed drug. So that's something like that's an inside information that would be useful for the series development as well, just to see. But I will get back to this probably in the next meeting to see what we have decided on this.
Okay.
Also, just a quick reminder. So DM52/1 was potent, gave very good potency results. So the benzene ring, the phenyl ring is of quite interest. So the next step would be maybe modification. So that's also the reason why oxyconazole analog would be of interest, personally, to me. And DM81 is piperonil. Something that's found in some antifungals as well. I'm doing it this week, so this will be included in the next batch. But it's I just started the synthesis of this, and that's it for UCL update for today.
What are the log Bs of these molecules?
I haven't included them, but they're in most of them have been calculated by Ben Perry. The ones that have been tested for sure, I do have them. It's just I haven't included them in the scheme, but they are in the range in our 3.5, 3.2, something like that.
Because according to the in vivo thing we're talking about, for in vivo efficacy, we think that less than 2.5 is needed, right? So you know, we need to keep that in mind if we're going to use it as a design criterion for new molecules.
It's not always been the case, so the pattern is not absolutely clear out there. Sometimes like compounds with higher log Ds were like quite potent. Because if you remember, we proposed that it's ideally 0 less than or equal to 2.5, but there are some exceptions to this rule based on the assays that we've done before. Yeah, but we can come back to this. But I have the log D values because Ben Perry calculated for most of them, especially for those that are yet to be tested. I think we have them. He just did it like recently, like a few weeks ago, like 3 weeks ago, I guess.
Yeah, the last batch I sent for calculations was in October, halfway October. And those were all added in the GitHub data sheet. Okay, then due to the sake of time, shall we move to the next items? The next outstanding item from the previous meeting was an item for Matt. It was to pursue the metal complex samples from COAT and report at the next meeting.
No, not done. I'll carry it over.
Okay. So I saw that Ma did upload the biological information on the spreadsheet. Ma, we didn't have further updates currently because you were busy with the finalizing of the data.
Yes. I worked with data analysis this month and prepare chemical list for the next in vitro test. So we are now updated this month.
Okay. So I tried to, the next item was that I would ask Ben to perform a statistical analysis on the Fennery mold data we had so far. Just a quick recap. We did in vivo efficacy for the Fennery moles. We found an association between log D and percentage survival in the larvae, so in vivo survival. But the simple statistical method I was able to do was simply a Mann-Whitney test. So I divided the percentage survival of all the compounds with the log D of 2.5 and below on one hand, and all the survival data of compounds with the log D above 2.5 on the other hand, and did a comparison. Based on Mann-Whitney, that difference was significant, but in the previous paper, the first paper, we did more sophisticated analysis, and I don't know those actually, how to perform those. So that was the question I was going to ask Ben. I sent Louisa this morning the manuscript and the data, how it looks like, and maybe she has some more information or more knowledge onto how to analyze these data in a different statistical manner.
Unfortunately, Wendy, I cannot help you on that. I am an organic synthetic chemist, not good for statistical analysis. I can still contact Ben and see if he had done any of this. Although I think his last month at the NDI was really busy, so I'm not sure how is he handling this. But I can have a look at the data and see if I can find someone within the NDI to help with this analysis.
Yes, that would be great. Or if you know somebody somewhere
I'll do
else,
that.
Yeah.
Okay.
Because unfortunately for me at Erasmus MC, I wouldn't be able to spot now who's able to do these kinds of analysis, but I can contact the statistical department to see if they can help.
Okay.
But I will wait on your feedback first, and then if you can't find someone, then I will continue from this side.
Okay.
Okay, and then the last outstanding item from the previous meeting was for Hung. on the manuscript. And maybe if we could do the same as what we did with Ma and Dimitri, that you could talk about the outstanding item and give an update from your side as well, then that will probably be the most efficient way.
Yeah, so for the manuscript draft, I have addressed all of the comments that was addressed to me or that I could resolve. So all of that has been resolved and updated. So I've also put together the supporting information for the SAR. So all of the figures that needs to go into the supporting information for SAR are now in one document. I've also uploaded the citation list for the SAR. So I actually haven't unified them into the actual articles just because we haven't finalized it. So because there was some issue with duplication of citation because we kind of just use different things and we put different bibliography software. So it's kind of messed it up a little bit. So I haven't actually put mine in, but I've got the list and I also got the citation to match that with the separate file so we can unify that at a later stage. Yeah, I think that's about it. So that part is done from me. Not sure what we're gonna do next in terms of like what we can do to progress the draft, I guess. I guess that's up for discussion for today. Also will be shipping my next batch of compound this week. So I've just got, I need to get them back from the NMR facility so I can actually dry them and ship, get a portion and ship them. It was a little bit delayed because the facility was busy, but yeah, I'll try to get that done this week. And I think there's also some series 2 from the Breaking Good project as well that should be coming with my shipment as well. But I will talk to Clementine and we'll organize that for this week.
Yeah, I'm leaving to Sudan for tomorrow, so I will be away 1.5 weeks. But Ma is in the lab, so it's best to contact Ma as well if you're going to do a shipment during the time I'm in Sudan.
Yeah, can I address that to you, or do I just give it like the name to Ma instead?
You could use my name on the parcel, but I'm not sure if I will have internet because I'm also going to the field. So
okay.
For the communication itself, it's easier to go via Ma, but on the parcel, you could use my name together with Ma's name.
Okay, yeah. And I've also claimed a couple of the MyOS code in the master list for my batch of shipment, but I haven't actually fully updated all of the data yet. I will do that as I do my shipments as well. So that should be done hopefully by the end of this week as well.
Okay, with regards to the manuscript, I updated the last survival data in the manuscript, updated the graphs which I made on the survival percentage versus log D. And I double checked the number of compounds screened in vitro if those were these were up to date, and according to me they were. So I did do those double checkings. But yeah, that's how far I went. So maybe it's now good also for Matt to go through it to see, because it's in a chemical journal. So I don't see much. Yeah, it's more your expertise than mine.
Yeah, happy to. So we're sure that Peter Rutledge isn't looking at it, Alice isn't looking at it, so it's good to go?
Yeah, so Peter's already looked at it in end of October, so it is ready for you to have a look at.
Great. All right, I'll do that. What's about how about the status of the SI document? Is it good to go?
Yes, so I've already put together in the manuscript itself, there is some of the data that I think shouldn't go into the main articles or the main body because it's just too long. So some of the I can show you actually. Just give me a moment.
I mean, just if we submit it tomorrow, is the SI document with the chemical data in it ready to send?
I still need to upload the NMR data.
So right now
For
we
my
have
side.
the main document itself, which is the main draft, which is this document here is on version 6. I've got the NMR spectra, which is one of the supporting documents. All of the spectra should be here.
Right.
Sorry. And I've also have this one as well. So these are some of the figures that are referenced in the body of the text, but I did not include it because it just takes too much space. As well as I think that's it. The citation, but yeah, that can goes onto the body later. So I'm not sure how to do this. So do we combine all of this together, or do we leave them as separate SI documents?
Well, ultimately, if it's a Word file, it can go all together. And the PowerPoint file for the NMR spectra could probably be left as it is, I suspect, because it can be converted to a PDF, and that'll be okay. Yep. But the data that includes all of the characterization, needs to just be ready for all the compounds that are in the paper.
Yes.
And for
so
any
the
compound,
characterization data are currently in the main body and not in an SI file. It's not in a separate file.
It's in the main body of the paper.
Yeah, it hasn't been taken out of the main body yet.
Okay, that's all right, but just as long as it's ready to go, right? So that we're not waiting on any carbons or high-res mass specs or anything.
Yeah, so that should be ready. I didn't notice that you were commenting on the consistency of the decimal places in, I think it was in Dimitri's part, but I read that, and I also make sure that whatever I've done is also consistent with what you're requesting up there as well. So that's also been fixed.
All right, great. So I mean, we just got to make sure it's ready to go, so that when we submit it, that's ready to go at the same time as the paper's ready. And also because it's an open science paper, we need to have the NMR data files submittable, right? So that people can reanalyze it. So otherwise it's like a regular paper, but this is meant to be an open science thing. So if for any compound that's in the paper where we provide the NMR data as a peak listing, we need the NMR data ready to submit as a separate file, right, for compounds, so that people can see the raw data.
So is that going to be a zip file, or how?
Yeah, something like that. It's likely to have to be on a university repository because it'll be too big, I suspect. But we just need to make sure that they're kind of ready to go.
Yeah, because right now all of the data are on my lab archives anyway, which is open source, which is open to the public. There's also an RDS system that is currently introduced, but I don't have a lot of knowledge on it because I haven't actually used it before. But if one thing that we could do is we can link that to my lab archives, which should have all of the data there, because I have spectra and also I have the raw file itself as well.
Yeah, that's good. So what we do is we make an offline copy of the lab notebook, so it's downloadable, and then you can post that on the university repository. We did that with the previous papers, and that's important, and I think that's useful. But I think it helps reviewers and people who read the paper in the future to have somewhere all the NMR files that are distilled out.
Same for UCL lab archives.
Yeah. So there's a list of all the NMR files, right, where people can get at them and they can reinterpret the NMRs, which is very, very useful. And then there's separate offline copies of lab notebooks that talk about the different experiments. And then our paper is distinctive. Yeah. So if you can be working on that while we're finishing the manuscript so that we're ready to submit all that stuff, that would be great.
Okay, Hang, are there other updates?
That's all from me.
Then we can go on with discussions points for this meeting. There are only 2 points left. The first one was Rowan's contribution of compounds from the Griffith University. So that was to be discussed. So maybe Rowan, you could introduce the subject.
Yep, sure. So I met Hung and I think it was Alison, was it Hung, that at the RACI National Congress a couple of months ago, and saw their presentation on this mycetoma project. So got chatting, and then Hung reached out to me again a couple of months later. I talked about a pure compound library that was predominantly natural products, and he said, that sounds like a good thing. Maybe we should get that tested. And so I sent an SDF to, I think it was Alison, Hung, and Peter. Matt, did you get a copy of that as well?
I can't remember, sorry. Probably did, yeah.
That's all right. Anyway, I just showed them, I declared what the library was. It's most of these compounds, Wendy, have been previously published. I think there's 3 or 4 that are in manuscripts at the moment that are new still, but everything else, these are known natural products. But I was just wondering if natural products could play a role in your particular project. And I saw Dimitri presented some natural product hits just earlier in the meeting. So I think I don't know what was the 512 compounds, which are in my pure compound library. The SDF was going to be triaged somehow, but I don't know how you were going to do that. And that's where I'm at the moment. So the compounds are available. You can screen all 512 of them if you want. They're in Compounds Australia, which is a national compound repository here at Griffith. They're in DMSO as 5 millimolar stock solutions. They can be plated out in any format you want, in duplicate, triplicate, etc., 1 microliter, 2 microliters, and then sent wherever they need to go. Initially, I thought this screening was done in Sydney, and so it was going to be a cheap proposition for me to ship the plates, but I've since learnt that the screening is done in Netherlands. That's correct?
Yes, unfortunately, we're the only lab in the world who's currently able to screen, so.
Yeah. So I think that the problem there is that, while I get discounted rates for plating of my library into 384 or 96 well plates, the shipping of DMSO on dry ice is a very expensive proposition. So I wasn't sure if Hung's idea was to, with Peter, look at the structures and do some sort of structure matching or virtual docking to pull out maybe the 10 best candidates. And then I could send you, Wendy, dry stocks, half a mig to a mig of those available and abundant compounds. And then you make up the stock solutions your end, because for me to send dry compounds to the Netherlands will cost me $300. To send the DMSO plates will cost $3,000 to $4,000. So I just don't have that budget, unfortunately. The second budget. It's funds are tight.
Yeah, we screened MMV libraries, and in the past we also got DMSO stocks, but their last library was also a dried format, but I'm not sure how difficult it is technically just to spot and dry them. Probably it needs to be under certain conditions that the compound is dried.
Yeah, a lot of the time we'll just send so we've got a 5 decimal place balance in the lab. We weigh the powders out, or the gums on this balance, and we send 2 ml glass vials with screw cap vials that are labeled. And then I do the calculations. If you want 50 millimolar solutions, I do that, and then you just resuspend in DMSO your end. And we've had no issues with that. My library's been sent to 20 other collaborators around the world and screened in lots of different assays, but never for antifungal or this mycetoma that I'm still learning about. So I just thought it might be another way of getting the library out there. It's the Davis Open Access Compound Library. So you guys you're all embracing open access. Why not join the party? But yeah, look, if Peter or Matt looked at my compounds we use Data Warrior to analyze structures. If someone send me an SDF, that's how I'd view the structures. If there's a certain motif that Peter, Hung, or Matt are after, maybe that's a way of reducing the workload as well.
Are you high throughput, Wendy, or it's really only a 96 plate a week sort of proposition?
Well, we can screen 384 wells. For another project, we screened 6,000 compounds using luciferase.
Okay.
So that assay is working, but I didn't go beyond that. So I didn't go to 1,600 something. So 384 is the smallest volume we went.
Yeah. Okay. Well, 384 is pretty standard these days. So most of my collaborators, that's how my library gets shipped out, and you know that's only 2 plates, depending on whether you want duplicates or not. But I think it's the cost of shipping that's going to be the bottleneck there.
So the cost you mentioned of the $3,000 to $4,000, is that the shipping cost, or is that also factoring in the fact that someone's got to actually plate these things out?
That would be the plating and probably the shipping. But I'd need to get a quote, Matt, from Compounds Australia, and they can give me an updated quote. But being a Griffith Uni employee, I get discounted plating rates. But World Courier, they give us a small discount because Compounds Australia send their libraries all over the place. But it's still, yeah, it's probably the most expensive part of the whole program is the shipping.
Yeah, I can understand. And unfortunately, I cannot afford the 3,000 euros either to just reimburse you the shipping.
Yeah, yeah.
But we
I have
did
a remark.
screen for methyl compounds. So.
I have a remark, sorry. I think you have published this paper about the antiparasitic activities of your natural products, and you have identified some 10, 15 active compounds. So maybe it's possible to concentrate at first on these few active natural products, to send them as powder to Wendy's lab in Rotterdam, then to see if they are active, and maybe we can decide then to go on with more natural products from your lab.
Yeah, that sounds reasonable. And I think in our earlier discussions, I talked about how the library, I've been working in the malaria area for some time, and I think it was the malaria compounds from my library that was of interest to Hung and Alison initially when I met them. So yeah, that seems reasonable. That will reduce the costs, and yeah, we can take it from there. How many, there's no point sending known anti-malarial drugs, right? You would have screened this program would have screened those. Is that right?
Well, we screened MMV library, so there are open boxes,
yeah,
but not the first one. So not the malaria box, because that was already no longer available when we started. So we started with the pathogen box. That was the first box we screened.
Yeah.
In the past, I did screen tea tree oil from Thomas Riley. So that's also an Australian compound, of course.
Mm-hmm.
And that had activity as well.
Okay. So I guess my question is things like artemisinin and all its analogues.
Artemisinin itself has been screened.
Yeah, mefloquine and those sort of things, because I've got some synthetic drugs in the library as well. They're probably 90 to 95% natural products, and there's a few synthetics due to my interest in anti-malarials. We were making some semi-synthetic, or we were doing some late stage functionalization chemistry on some of these known malaria drugs. So maybe not send those. Is that what I'm hearing?
Yeah, so artemisinin itself has been tested.
Yep.
It did have very minor activity, I think. Tea tree oil was much more potent. Are there also siderophores in your library?
Yes, there are, but they've got a bit of a nasty. Most of my siderophores have got catechol units, which are a bit of a no-no for drug discovery. But I don't know how. I saw some structures presented today that look like they've got chemical alerts as well that you've highlighted green, which I was surprised by. It's not as much of a concern for this project?
Well, our main concern is how to get things into the grain itself. So in the hyphae, so in vitro, when we look at the fungal hyphae, much more potency is found, but in real life, the fungus will form a grain, which is like a 3D biofilm, which makes it very difficult to penetrate. So what we did with another collaborator, we did do transcriptomic analysis, and we found that many siderophores are actually secreted during the grain formation. So if those siderophores are secreted to obtain iron, that means that they are also transported back into the grain itself. So potentially those siderophores could be either an antifungal by themselves or equipped as a carrier to gain entry into the grain, because that's the tricky part, and that's what we're trying to solve with the log-D, to see what is needed to penetrate the grain. But you could also think of it the other way around. What can we add to the molecule as a carrier to get drugs in? And then a siderophore might be potentially something we could think of.
Okay, all right. All right. So I think Bernard's, will I run with Bernard's idea and pick out, so Bernard, was that the paper with Vicky Avery where we were looking at leech and other anti-infective? I think it was a molecules publication from 2017. Is it Bernard? Sorry, I might have misremembered the name.
Not sure. It'd be good to get the link to that paper just for the minute so that we can have a look at it. If someone could send it along, that'd be great. Because I think that's a good halfway house. Unless, you know, DNDI or ASI want to stump up, you know, $4,000 for you, Rohanna. You know,
there was another paper that was published by DMDI where they were looking at aminothiazoles, which were the ones that you were also had as a series. I posted that in series 2, I think. That was an interesting paper.
Then the test of a subset would be a good idea in the interim. Yeah. Okay,
so yeah, I think it's good to include that paper, Bernard, to send it in either on GitHub or at the chat at the Zoom. Due to the sake of time, I would like to go to the last discussion point, and that's the crowdsourcing update. There were incoming contributions from different schools. It is an action point for Kimberly, but maybe someone else knows something about this.
Yeah, I can take this. So there's some contributions incoming from a school in the UK where they want to make molecules, as well as the existing school 7X. So there's 2 or 3 schools now, and there's a group at York who want to get involved in making some compounds. And I guess I'm kind of directing them to the series 2 or series 5. I forget which one it is. The one that Kimberly and Hung have been doing with the school workshops, because the chemistry is quite nicely worked out. What I'm going to do is post something as a comment in the current issue, which is intended to be text that summarizes for any potential new groups who want to make molecules for that series, what they need to do in order to be able to contribute. So what they need and what they need to be able to do.
Is this the aminothiazole series? I found a paper on that. DNDI just published a paper in 2022 about these things as being antifilarial.
Yeah,
and they're very similar to the mycetoma compounds. There's probably some cross-reactivity.
There's a reason for that. Yeah. So just to make sure that we know what the synthetic route is that we're recommending, and that we know what it is we're asking people to do. So I'll post something below the issue, and if people can comment on that and make sure it's okay, then we can direct people to that. So if we get a request from someone to be part of MycetOS, but there aren't that many chemical resources available for contributing that we can tell people, well, you can do this, and this is the thing that's currently needed. So because I've got one school asking me which molecule they should buy as the variant point in the structure, and I want to make sure that we're all on the same page. So I'll do that. I'll ping everyone, and I'll ping Kimberly, and just make sure it's right.
So this DNDi paper where they made these aminothiazoles, could we ask them for samples? Because there's a couple structurally diverse ones in there in that ACS publication.
Yeah, my expectation would be they're already made, right? Already made and tested.
Is that the JMedChem one? Sorry.
Yeah,
Can
filarial.
Okay. Okay, so that's, who is that? Van. Okay. I can ask anyway to see if who synthesize and who are they, if they are in our inventory. If they are in our inventory in India, we can definitely ship some representative compounds to see if there is any correlation. But just maybe you can write me, Anthony, and then we can discuss maybe if you can pick a few compounds, then I can ask them.
Maybe, yeah.
Is that okay?
Yeah, that's fine. I don't have I do have full access to this paper. It's open access. Okay, I didn't know.
Okay.
Okay, thank you. Yeah.
Nice.
And maybe it's good to discuss that also on GitHub so that it's in the open space.
Yeah, definitely.
So what
Okay,
kind of
yes. Sorry, you can write me on GitHub then.
Yeah.
Okay.
Okay, since we're already 10 past the hour, are there any other businesses we need to address during this meeting?
I just have a quick question that I was just hearing you guys, I think it was Dimitri talking about log P and log D. Is that usual spend stuff to do for you? Does he usually calculate log P or what kind of program do you use for that?
Ben start drop and Ben
okay.
for this.
Okay,
and it's quite important
guys...
for consistency because
absolutely. No, you can.
We calculate using different software, then it's not going to be consistent, and there will be differences like for the same compounds. That's what he told at the beginning of the project. So
yeah, so what I usually did, I sent him via GitHub and via email usually a list. If there's a list pending, then I will simply drop that, and he copy pasted, I think, in the program, because he usually replies within 5 minutes.
Yeah, yeah, that's usually it. We can open a CDF file, so we just click a button, log P, and then export as a CDF again. So
that's why it's a bit simple. Like sometimes difficult when planning new molecules. Like for example, earlier today, Matt asked, like, do you know the log D? For those that he was asking for, yes, but they came up like just now. But at the time of so we use like different software, but like it's a bit like difficult sometimes. Because the numbers vary, so start drop would be the best option, obviously.
Okay, no, fair enough. I can continue to do that.
Thank you.
No worries.
Any other businesses? Then, if not, then thank you all for the nice discussions and contributions, and I hope to see you all next month.
January 24th, I reckon.
I'm just going to look in the agenda because it's now, that probably the next one is in the middle of Christmas, isn't it?
Yeah.
So then it's best to skip the one from December and
I suggest Boxing Day.
Sorry?
I suggest Boxing Day after Christmas.
Yeah, we in the Netherlands, we have 2 Christmas days, so that's our second Christmas day. No, but then let's do the one in January. So I will, Matt, will you put that also in the minutes so that it's clear for everyone that we next meet in January?
Yeah, will do.
Okay, then thank you all and see you next time.
Thank you.
Great, thanks.
Thank you, bye-bye.
Bye, everyone.
Bye, thank you.
Bye-bye.
See you.